Okay, I think it's the time. We are going to start with our last session from today. It's going to be led by Frédéric Yayan. He's a territorial manager in Averion Instrument. He's working there since 2018. And before, he has developed a long career as a researcher, so he had a strong technical expertise in AFM imaging and also in optical microscopy. I would like just to remind all of you that we have a chat. Then if you wanted to share any comment or any question, please do it there. So, Frédéric, the stage is yours.
Thanks a lot. So I will start sharing my presentation. I think now it is shared. Yeah. Okay.
It is.
Okay, it's excellent. So now I'll go full screen and welcome everybody for my talk entitled Extending the Domain of Super Resolution Microscopy. I am part of Averion Instruments since a little bit. Since a while now, and I'm together with our partner IZASA Scientific, taking care of Averion Instruments in Spain and Portugal. And I would like to show you today how the domain of super resolution microscopy is being extended by the production of cutting-edge commercial machines, and how it's going to be extended both in the sense of hardware and usability improvements of our systems. So this is the outline of my talk. So I will start introducing the company. I will talk a bit about some confocal aspects that we are generally not talking so much about at Averion Instruments, given the history of our company in super resolution. Then describe the history of STED, how we developed it into very simple commercial instruments, such as Steadycon, and then facility line or infinity line. Talking a little bit about dyes, because there are often questions about the domain of applications of certain dyes to STED microscopy, and finishing by introducing the newborn of Averion Instruments, which is Flux, an invention of Stefan Hell that was successfully transferred to Averion Instruments into a commercial product. So the company history starts in 2012 with 7 members of MPI in biophysical chemistry in Göttingen. So you can maybe recognize here Stefan Hell, who's the laureate for a Nobel Prize for chemistry in 2014 for the invention of STED microscopy, among many other improvements of super resolution microscopy that he's done. So basically, the company it's really the inventors of STED, actually, making commercial STED for everybody and transferring really the top-notch technology of super resolution and confocal into good commercial instruments. So the company is still fairly young. I mean, it's growing in spite of the corona crisis. It is still growing, and we are still releasing new modes, and I think the motto really is R&D. It's a very R&D based company, as you will see later. More than 50% of the staff has a PhD, 35 years of age on average. I'm a little bit older than that. There are a lot of publications coming from Averion Instruments, microscopes, of course, but the laboratory is itself also publishing a few development papers a year. We've opened a branch in the US. We're, of course, teaming with IZASA in Spain and Portugal. And as I said, what characterizes us is really the pace of development of new instruments and new modalities, which are generally very original ones, things that were never done before. And we can actually count something like maybe 3 new releases every year. So you can see, for example, in 2019, we've had the release of a new instrument in facility line, some new modalities of in-depth imaging in adaptive optics, and then Flux, which is the highest resolution fluorescence microscopy that exists on the market at the moment. But of course, I mean, to understand the value of super resolution microscopy, we have to put this in the context of confocal microscopy. And it is often overlooked, but all Haber state systems are fully fledged confocal microscopes. And I really need to insist on that. I mean, it starts from a base of a confocal microscope, and all the instruments of the state family of Haber instruments are indeed actually to start with confocal microscopes. So the Steadycon is really the entry-level compact line that does 4-color confocal, 2-color 2D stead. It is an add-on to any inverted or upright microscope, and very easy to use. And as I will show, actually, our aim was to take the lessons from the Steadycon, which was really a very successful machine and still is a very successful system, and make really a cutting-edge instrument that also has a very, very big simplicity of use, so that would actually bring 3-color 2D stead and 3D stead for everybody. And here we have this facility line, which is, as a matter of fact, also a confocal microscope. And next to facility line, we have here infinity line, which has actually the same sort of properties, the same level of performance as facility line. The difference being that the optical bench is larger, and that introduces actually some very nice capabilities of customization. So for example, if you want to do something really special, or let's say if you want to have like up to 8 excitation lines, I mean, we of course do not know what would be the purpose of that, but let's say if you want to have a whole lot of lasers, then you have a lot of space in that optical bench to do really whatever you want. But in the same time, infinity line can replicate what facility line does, and you can also have a very easy to use system for your platform. So we often talk, being the offscreen of the lab where state super resolution was created, we often emphasize the performance of instruments in state super resolution, but it's a little bit the tree that hides the forest of confocal features that I'm going to detail here a little bit, but not too long, of course. I mean, this is a super resolution session. So among other features, what is really common to all Haber systems is going to be a robust alignment, being the aligned by design features of the Steadycon, or motorized alignment, automatic alignment for facility and infinity. Then you, of course, have motorized pinhole selection, otherwise it would not be very easy to use. You have the possibility to control the motorized stage and perform mosaic imaging and multi-position now on all our microscopes. All our microscopes also have a Z-drift compensation, which goes all the way to autofocus on our larger systems. We have multiple excitation channels, multiple excitation lines. Most often, they are based on pulsed excitation sources. We can perform flame upgrades. All our systems have really highly sensitive gated FED detection, which are based on avalanche photodiode working in photon counting modes, and they are the most sensitive of the market. And of course, we have a few different barrier-specific modes that I'm going to detail in the presentation. And so, of course, our microscopes, they are super resolution, but they are also excellent confocal microscopes if you want to do really large-scale imaging. So here, this is a tile and stitch image of Drosophila testis multicolor that was acquired on our facility line. So you can really see the kind of things you can do on large samples as well, or on plant samples. As a matter of fact, this is a ZMI's kernel. This is a section of a corn, actually, kernel imaged in multicolor with a Steadycon and then stitched, so you can see the image is more than a millimeter width here. So of course, confocal microscopy has a broad range of applications, and we want to make sure we bring as many options as possible for people who perform it. But confocal microscopy is itself not without limitations, and that's the reason to be of super resolution microscopy. If we look at those nuclear pores here stained in 2 colors, then it's really, really actually hard to distinguish any sort of structure. And if you would actually do some electron microscopy on it, then that's what you would see. You would expect actually to see a ring. This is a pore, a nuclear pore. And the ring would have dimensions of a little bit more than 120 or 130 nanometers diameter. So of course, I mean, many people who attended Spawn probably saw many explanations on the diffraction barrier, but I still felt like I had to add an explanation here. So as you are actually focusing a collimated beam through a lens, then the converging beams are going to create an interference pattern due to the wave-like nature of light. And the zero order of this diffraction pattern, as a matter of fact, is going to be a diffraction limited spot, which dimensions relate directly to the wavelength of light and the numerical aperture of the objective that you are using. And so if you actually use visible light, which is often used for fluorescence, then we can estimate that the width of this diffraction limited spot is going to be 200 nanometers, and the actual spread is going to be 500 nanometers. So this is a limit that's going to be hard to circumvent, even if you try to use really exotic refractive indices. Then there will be issues after a while, or if you try to use different types of wavelengths. Of course, these are not the ways to go. And it's really important to be able to break this limit, simply because a lot of the life that we are all studying is happening on the supramolecular to molecular level, which scale is going from the nanometer to the 100 nanometer range. I mean, be it the diameter of microtubules, which is in the 25 nanometer range, or those, let's say, prokaryotic or eukaryotic ribosomes, which are a few tens nanometers across, or histones and so on, or even viruses. I mean, it's very fashionable these days, as you know. So that was the reason to invent super resolution microscopy. We had to understand the mechanisms of life on the supramolecular level, and really try to circumvent those limitations of optical photonic microscopy. And as you all know, a Nobel Prize was awarded in 2014 for the invention of the microscopes to three of the people who most contributed to its invention. And in the middle, we can here see Stefan Hell, who is the inventor of STED microscopy, featured here. And you can actually find those nuclear pores on the left side, here imaged in confocal, and here in the middle and on the right instead. And if you do a close-up and look at the bottom panel, then you can see that the two colors that we could hardly resolve into a structure are now really very nicely resolved. And you can see actually that the red stained part is going to do a ring, and the central part can be stained and really separated from this part. So this is one of the first messages, actually. If you overcome the diffraction limit, then you can start doing structural analysis. Now, if you actually calculate the diameter of this pore and you divide it by the number of monomers into that ring, then you can guess that we are far below 50 nanometer resolution. And this was acquired actually on our instrument by our instruments staff, which is our head of applications. So the principle of STED microscopy is based once more on point scanning microscope, a confocal microscope. The excitation beam is going to excite molecules in the sample and send fluorophores to the excited state. Then a so-called quenching laser beam is going to send all the excited molecules in the periphery of this region back down to the ground state. So only the central region is going to be able to emit some fluorescence. So I'm not showing the details here, but in order to actually have that quenching, what we use is a process of stimulated emission. And to perform this stimulated emission, which is going to bring back the fluorophores from the excited state to the ground state, and to selectively just do it on the periphery, we use a donut-shaped beam of red light at relatively high power. And if we actually look at those 20 nanometer particles, which were not really resolved at 20 nanometer objects, I mean, you can measure the full width at half maximum of 221 nanometers here. Then if you start adding that donut-shaped stimulated emission depletion beam, that STED beam, then you can reduce the region of emitting fluorophores down to 132 nanometers. And as you increase the STED power, the region of effective emitters at the center of the donut is going to shrink until you're actually able to reach the point that the region, the central region, is matching the dimension of the particles. So you can actually tune the spatial resolution by tuning the power of stead. And there is a very important message here, in the sense that I think in the beginning it was often said that stead really is burning samples, but that's not actually close to the truth. In reality, you only need really high stead power when you need to go to really very high resolution. If you look at the shape of this curve, you can see that the relation between the increase in resolution, the resolution enhancement factor, and the stead power is nonlinear. It's actually related to the inverse of the square root of a stead power. And so if you add a little bit of stead power, then what happens is that you get really a very drastic enhancement of resolution. So you don't really need much stead power. And this situation is even further improved if you use pulsed stead. Yes. So of course, we are needing here actually some dyes which are going to be sensitive to this process of stimulated emission. This is a photophysical process, and we need to have dyes with certain properties. The properties of the dyes that we are going to use are going to be, first of all, related to the wavelength of a stimulated emission beam that we are going to use. Here, actually, if we use a 775 nanometer beam, then we need to have fluorophores which have emission spectrum intersecting that wavelength of stimulated emission depletion. So here, actually, we have Abair Star Red from our sister company Abair, which produces the dyes and tests them on our instruments. And we can see that this fluorophore fulfills this condition, as well as Abair Star Orange. We can see, however, that the extent of this intersection is going to be less. And we could guess, actually, that Star Orange is not going to be as efficient as Star Red, and it is actually verified in experience. We also want to avoid that the stead beam re-excites the fluorophore, so we want to have quite some distance between the excitation maximum and the stead beam. So then the next criterion is, of course, very empirical, and fortunately, Abair is selecting really the best candidates as stead dye, and we actually now are able to use 3 very good stead dyes to do very nice high-resolution 3-color stead using Star Red, Star Orange, and Star Green. And currently, Abair is releasing actually some new, brand new live dyes. I can show you an example here. Those dyes are really bright, photostable, which is a very good, very important condition instead. And it's really recommended in Confocal, and it's not anymore coming soon, of course. I mean, those dyes are released. But now you can see a very nice stead, live stead image using Abair live dyes. So you can actually do live stead now. It's possible using those dyes, both on cells and tissues in Confocal and in stead. So one of the avenues that Abair took in trying to democratize stead and extend the domain of applications of stead was to release the Steadycon. I'm not going to talk too much about the Steadycon because you have many people have seen it last year at SPAUM already. It's a very nice compact 2-color stead setup, 4-color confocal that gives you 50 nanometers or even less. It can go down to 35 nanometers resolution within a few clicks. And this works as an add-on to any microscope, be them upright or inverted microscope. So you can see here the combination of the Steadycon and upright microscopes. The Nikon setup is not mentioned here, but if you've seen the IZASA demo last year, you know that the IZASA demo system is actually on a Nikon upright microscope. Now, it works also on an inverted microscope, and you can actually plug it really to one of the side ports. So the condition for that is you only need a side port with 100% transmission and no lens. So really you need a 0-100 port. no lens, and then it's really going to work fine. And of course, I'm not mentioning the features that I mentioned earlier, but all our microscopes are equipped with high sensitivity avalanche photodiodes. They are all this is equipped also with a Z-drift compensation feature. It can have a flame upgrade. And just to prove actually how easy this system is to assemble and to use, actually to make it work is really easy. I actually could move it during lockdown, and actually it's still in my bedroom right now. I can perform experiments. The cat doesn't mind actually, so Sylvester is sleeping here, and I'm acquiring actually those brilliant images, reproducing part of that Nobel Prize poster, you know, with that staining of nuclear pores, in which I can really distinguish and count nuclear pores within a nucleus at 65 nanometers resolution. And here we have a very nice Golgi staining with star range. And a little bit later, actually, the cat is getting up. It's later at night. After 5 samples, I actually switched to neurons. And here, actually, we can really resolve on those neurons the alternating green and red color of beta-3 spectrin and adenosine. Here is a little bit closer. So this is compared to confocal imaging. This is raw data. The confocal image cannot resolve any structure, but thanks to the resolution of STED, we can actually see really clearly that periodic assembly of spectrin within neurons. So if we look now at Golgi in confocal instead, so in confocal it seems like you see a boundary actually between the red and the green domains. In reality, STED refines quite a lot the situation, and not only can you start counting clusters, but you can also see a bit more clearly which color is, who is close to who, so you can start seeing actually red spots closer to green spots that you couldn't see. So the message here is that you could imagine that STED helps you count objects. It can help you structurally characterize, refine your colocalization. This is here in the bacteria. This is a HTRA, which is a serine protease. And right now, actually, you could not really localize this protein. And you are able to localize and even possibly count and analyze the distribution of this protein within your bacteria. And this is really how STED really helps. I mean, it resolves what confocal cannot. Cannot see in the sense that you need a signal, of course. So what confocal cannot resolve can be resolved by STED. It improves a lot the colocalization analysis that you will want to do. And I encourage anybody who struggles a bit with the colocalization or to switch to STED, and maybe you get a different result, especially, of course, when molecules are really close to one another. It enables structural analysis of biomolecules. You can count biomolecular complexes or molecules. You can measure objects. You can start doing a lot of things that you couldn't do with confocal. So now, coming to our higher-end systems, facility and infinity line. Of course, I've shown only 2D STED, which is what Steadycon does really well. But life is, of course, made of 3-dimensional assemblies as well. And if you are interested also in questions that involve, let's say, tissues, if you want to do more than 2 colors or 3 colors, or if you want to really go further with your STED measurements, then we wanted actually to have the same sort of ease of use as we have with the Steadycon, and the full-blown capacity of a STED, which goes really far, as I will show. So here you can see a panel of samples that we wanted to make accessible to most people, from Drosophila testis to inner hair cells of mice, neuromuscular junction of Drosophila larvae. This is an expansion microscopy experiment. So these are centrioles expanded and imaged in 3D STED on our setup. This is published actually by the group of Paul Guichard in Switzerland, and so on. So that's the reason why Facility Line was released last year. We wanted really to have a high-end super resolution microscope, which would actually have all the features also of a good confocal microscope for a facility. So we wanted it to have really unprecedented user-friendliness. And to give you a hint of a level of performance, this system actually gives you free color 2D stead down to a 25 nanometer resolution. And we can use the easy 3D stead of our microscope, which I will explain a little bit later, to get less than 75 nanometer resolution and down to a 60 nanometers isotropic resolution in XYZ. So adding up to all the features that I mentioned earlier on facility and infinity line. So of course we have 2D and 3D stead. We have all the features, robust alignment, motorized pinhole selection, motorized stage control, autofocus. And we have actually unique properties that you can only find on facility and infinity line. So we add up actually some very high resolution spectral filtering based on a couple of notch filters that overlap to create a tunable spectral window. Adaptive illumination, which I will explain during the talk, which helps managing the dose instead and brings instead to capabilities of live imaging, and also helps a lot the reconstruction of volumes at really high resolution. We introduced a new matrix detector, which is an array of avalanche photodiodes, which I'm going to talk about a little bit, and which improves both the signal to background ratio in our measurements and also improves optical sectioning. And adaptive optics, which is very important here if we want to bring both confocal and 2D and 3D steady imagings to depths of several tens of microns down to 100 microns. So now, of course, our systems are equipped with multiple light sources. We can have up to 5 with facility line. We can have 4 or 5 nanometers continuous wave or pulsed to see the nucleus. Then we will be able to do confocal and 2D and 3D stead using red sources in combination with a 775 nanometer pulsed stead laser. And we would be able to image fluorescent proteins like GFP, YFP, or organic fluorophores, fluorochromes like Oregon green or Abyr star green in combination with a 595 nanometer stead pulses, which gives actually the system at a minimum a 3 color, at a minimum 2 color or 3 if you had the 595 laser. You can even imagine having a little bit more instead. We wanted the operation to be easy and stable, which is our way also to extend the domain of super resolution, just to make it really easy and stable and productive for everybody. So we introduced a push-button auto-alignment procedure, which is going to align all laser beams and pinhole within 90 seconds. So you come in the morning, you have an alignment sample, you click a button, and after a couple of minutes, you are ready to go. Then you place your sample, and we have very nice autofocus features, so you find the focus readily on your sample, and then you get going in confocal instead for hours and hours, so the operation is really stable. So I have to apologize right now to my company, but I really wanted to show the software, and I took that snapshot of this of the software. I don't even know if I have a right to do that, but I really wanted to do it. And this is actually how it looks like. So it's really, really a stripped down software interface with a lot of features, but it is made in such a way that it's not too much, it's not overloaded. I will not explain you in too much details, but really we took the lessons from the experience of the Steadycon, which is really a brilliantly easy to use system instead in confocal. And in principle, you can get 2D or 3D stats within just a few clicks. And this is an illustration of a workflow. So here we are going to be doing imaging of neurons in one color very briefly. Now focusing, then actually doing a tile scan on the sample and getting a large scale image, so we can actually explore our sample and select a region of interest, which is going to be done in a minute. So I think the movie is a little bit accelerated, I have to be honest with you. I'm not completely sure, but yeah, so we can select, for example, that tiny region of interest here on which my colleague is going to be doing some state acquisition. And you see he tunes the state power and the excitation power. And within just a couple of minutes after putting the sample on, he gets actually a steady image. So it's really, really that easy. And of course, we have upgrades such as the possibility to add film in confocal instead on those microscopes, but this is something I'm not going to be mentioning. We have software integration of Becker and Hickel and PicoQuant film. So we had the film card, we have an integration within our software, and it is possible to get an overlapped lifetime and fluorescence image. So yes, you get overlapped confocal lifetime image or stead lifetime image, and you can actually have a live reconstruction of those images too. So you can do that too with our system. Now coming to the results, of course, to this stead is going to give very good results. We are really happy to be seeing our systems in the world working hard on coronavirus and SARS-CoV-2 and trying to help push our knowledge forward. So as you know, those viruses are going to be really interesting for super resolution because their size is going to be in the 100 and 200 nanometer diameter range. So typically things that are going to be very hard to study with confocal due to the diffraction limit. And here we can see that the magenta stain of SARS-CoV-2 is going to be resolved really, really very nicely. So we can start seeing individual and count individual virus particles within those very characteristic protrusions that they produce on the cells that they infected. And this data was obtained in Montpellier by our colleagues Delphine Muriot, Ciel Favaret, Marie-Pierre Blanchard. Now, of course, with a system like Facility Line, we can obtain more than 2 colors. We can obtain 3 colors. So this is a confocal image. Again, we have nuclear pore staining, intermediate filaments. And I think those are peroxisms in the 3rd color. I'm not completely sure anymore. And here, very nice resolution enhancement in all 3 colors instead. Now, we wanted to add also a resolution enhancement in the 3rd dimension. And this is absolutely not to be overstated. The spread of the confocal point spread function, as I showed earlier, is 500 nanometers or even more if you actually start exciting in far red. So now, instead of those neuromuscular junctions of Drosophila, we can see that we go from a situation where the morphology of the species of interest is completely elongated to a situation where we actually go a factor 10 down in resolution, and we can really start seeing those neuromuscular junctions much, much more like they actually are, and not with that elongation, which is typical of confocal. So again, to illustrate that tenfold improvement in resolution, here we can see some slicing through a kidney tissue, which is labeled in 2 colors on podocin and nephrin. And whereas on the left, actually, it's really hard actually to separate the 2 colors. And also where you can see that those regions are really elongated. Here we can see, we can start identifying individual spherical point spread functions. So they are almost spherical 70 by 70 nanometer by 70 nanometers point spread functions. And this is really a huge improvement. Now, several features of our microscopes enable us to perform these 3D STED at really high depth and really high resolution. One of them is adaptive illumination, which I'm not going to talk about immediately. But the second one is actually adaptive optics. So we have a first level of adaptive optics on our STED microscopes, in the sense that our 3D STED is not made with a fixed faceplate or a fixed vortex. But we are using a spatial light modulator, which is going to be helping us to correct for spherical aberration. So here, actually, in that experiment, we were using an oil objective with refractive index of 1.52, whereas the mounting medium was here 1.45. So we're in a clear index mismatch situation. So we would expect actually both the confocal and the steady image to basically die out as we go in depth, as spherical aberrations start distorting the stead 3D donut, and as we start distorting also the excitation beam in confocal due to the index mismatch. But here, actually using a spatial light modulator, we are able to tune back the 3D stead donut to its original shape, and we're able to recover a resolution that's comparable to what we have on the cover slip. And you can see it actually on the right-hand side. That's here, the E caption represents what we see at 20 microns depth in 3D stead. And we can see now on the H part, which is at close to 70 micrometer depth, that we actually get really comparable resolution. Now, what we can see, however, is that the signal is dimmer. And as we go further and further in depth, we are going to lose the confocal excitation to spherical aberration. That's the problem that we have. And then again, spherical aberrations are not the only problem. I mean, we could actually end up with coma aberration or a lot of arbitrary issues. So I invite you to go watch the Aber Instruments summer symposia, which are on our webpage. There is a very nice adaptive optics webinar, which is going to explain actually the technique of adaptive optics using a deformable mirror. And this technique now enables us to correct actually both the confocal excitation and stead. And we are acting now both on the excitation and on the emission beam. And this is actually an illustration of how it works. It's really extremely powerful. So we have this collaboration with Albrecht Hase and Amélie Cabirol from University of Trento. I'm sorry, I didn't write the acknowledgements, but I have to mention that. So that was done. The sample, the B-brains were done by Albrecht and Amélie in Trento in Italy and provided to us. Those are cleared samples. And here, actually, whereas we can see that this image is very nicely defined at about 30 microns depth, we see actually the distortion precludes really a very good spatial resolution of a sample down to 135 microns depth. Using the deformable mirror to correct for spherical aberrations, among other kind of aberrations, optical aberrations, we are able actually to recover down to 100 microns depth a confocal image with definition comparable to what we have near the cover slip. So this is really, really a very big advantage here. Now, I'm not mentioning what we obtained in 2D stead, but the results were really spectacular down to a 90 micrometer depth. To adapt to the improvements that we could have in our, let's say, three-dimensional situation, we introduced a new detector, a matrix detector, which is going to be improving actually both confocal and stead. This matrix detector is an array of avalanche photodiodes, and those are actually going to act a little bit like multiple pinholes with a small aperture, so acting at sub-area unit in comparison, let's say, to a single detector with one pinhole. So in principle, we are going to be able to improve optical sectioning thanks to that. And we actually devised at Aber Instruments, my colleagues in R&D at least, devised a strategy to use those detectors to empirically determine the background on objects and perform a background removal routine, which we call actually differential imaging. So I'm going to illustrate that here. And so whereas in conventional stead, we get this image, those are actually GACO2 cells stained by actin, and we can see now those microvilli. And we can actually see here that the background is going to be much, much, much more reduced using that empirical determination of the background using the matrix detector. Now, coming to a confocal use of this differential imaging, here we have those cells stained with GFP spark fusion proteins. So we don't know which protein of interest, but we can see that the labeling is fairly widespread. And we see, if we come back to the start of the movie, that we have very extremely bright regions here, very densely labeled regions. And here we are working in a confocal with a pinhole at 1 area unit. Now, using the matrix detector to improve optical sectioning and backroom removal, what we see here is that we actually remove substantially the very bright regions, and we are able to see really what they hide, whereas it was not really possible in conventional confocal. So we have both background removal and improvement of optical sectioning. And here we can carry on doing that imaging actually for hours and hours in confocal microscopy, which is very nice. That's 280 minutes, so almost 5 hours of imaging. So that gives me a transition. I'm going to ask a small question. Would it be possible actually to do live imaging for hours and hours with STED, at least for a few hours? Would it be possible? I mean, we know STED is using a powerful light beam, and of course, you know, STED. We use pulse STED and pulse excitation, which are really excellent for keeping our samples alive. However, when this movie wants to stop, we want to be able to do even better than that, and we want to have as little illumination on the sample. What we want to do, as a matter of fact, is we want to illuminate the sample only where it's necessary. And here is a number of applications for what we call adaptive illumination at our instruments. So what the reasons why we want to manage the dose of illumination on our sample is, for example, if we want to do 3D STED. Let's say we used a pulse STED, which is already a very big improvement of a continuous wave STED. I could give you quantitative numbers. It's really a significant improvement. Now, when we do 3D STED, we always fear that we are going to bleach the sample before we finish the stack, because the STED light is going to be illuminating the upper and lower planes before we even image them. It could be actually the same in confocal microscopy. We could have a very light sensitive sample, and we want a way to manage the dose on those very light sensitive samples. Now we could have living cells that we want to be able to image at super resolution and keep imaging them for ages without having too much phototoxicity. We may want to optimize the use of our light dose to preserve the sample and go to the highest possible resolution, as on those DNA origamis. Or, as on the right-hand side, we may want to preserve the sample from being hit in parts which are really light sensitive, in the sense that they are going to damage the illumination is going to damage the sample. So this is here extracted from a scientific report publication where so-called guided state, which is adaptive illumination, was used to image the malaria invasion in red blood cells and avoid actually that yellow chunk of hemozoin, which is a metal cluster, which is going to cause destruction of a sample upon STED light illumination. So yes, what we want to do is reduce light exposure by adaptive illumination. So how we are going to do it? We are going to first find our cell and identify our substructures in confocal microscopy. Now, let's say we are scanning everywhere. What is happening is that the edge of the STED beam is touching the region of interest in plane, even before the central region has excited the fluorophores, and even before we have collected a single photon from that central region. And we can imagine very easily that the upper and lower planes are going to be illuminated. If STED is only turned on when the confocal is close to the regions of interest, then we already save the sample a lot in both the upper and lower planes, and even in plane, actually. So this is exactly what we do. And to select the regions of interest, we define a certain threshold of confocal intensity. And we can fine-tune that threshold manually. We have a first guess from the software, and then we can actually decide whether the contouring is right or not. And we are going to decide that the state is only going to be switched on when the light is overcoming that threshold, when the emission, the confocal emission, is going to be over a certain threshold. And the microscope is going to be taking that decision pixel by pixel. So within the region where the emission is going to be below a certain threshold, we have no stead. And then when we detect a certain emission count of photons, then we switch on the stead on that pixel and we start doing stead, and switch it back off when the emission goes below that threshold. It's a dynamic process. And here is the result of that procedure, which is called rescue. So this is how many frames that you could do here in 2D stead using rescue. And this is what we would do with conventional pulsed stead. So you would imagine actually that you would get maybe only a couple of frames if you would use continuous wave. Using pulsed stead already gives you the possibility to do multiple frames, successive frames, and using rescue helps you actually doing several frames in a row, up to 10 or more, without really suffering too much from bleaching. I mean, we do have some bleaching, but much less. And this is the open door now for living cell stead. So here is a confocal image, and here is a stead image. Now, what we can see here is that we obtained the steady image using rescue. So we used that thresholding property of rescue, and we only switched on the stead next to the regions that were contoured. And if we reconstitute the regions where the stead was switched on and off, we can see that we are actually seeing the structures really well, and without really selecting out regions of interest or not taking even too much, and still preserving your samples for that was a few hundreds of frames, and that was image for a few hours at the longest. Now, can we actually improve this? We can do that by improving the confocal throbbing mask. The reason for that is that the definition of this confocal throbbing mask is going to be limited by the diffraction limit, very simply. So the spatial resolution limit of confocal microscopy is going to be setting a limit as to the quality of the contouring of the regions of interest. And as a consequence, we can see actually that the stead beam is illuminating a sample where it shouldn't be illuminated. So we can use that property that a little bit of stead light is going to lead to a large increase of resolution and refine our mask, doing an intermediate stead step at lower resolution. And here, actually, if we calculate the light dose that we use on that sample, then it goes down by an order of magnitude. And the result is that we are going to be able, just as we are able to do some very nice 3D stead in confocal. We are going to be able to do some 3D reconstruction in confocal. We are going to be able to do some very nice 3D stead at very high resolution. Here we can see those dendritic spines extremely nicely resolved at very high resolution in 3D stead and without some bleaching. And this technique that I showed with intermediate stead step, it's called diamine. So the techniques are described in the literature. So I invite you to go and have a look at the literature on rescue or diamine, or learn more on that on the website, or go to the ABR Instruments Summer Symposium. Just a few comments on dyes, because I often have questions on fluorescence proteins. Fluorescent proteins may be used instead, but not all of them. We identify YFP and citrine or amnion green or GFP as very nice candidates in combination with 595 nanometer stead. Then more and more proteins are coming in the far red range to be used in combination with 775 nanometer stead. And we can actually start doing intravital microscopy as published actually here in scientific reports by the group of Catherine Willig. So this was not actually on aberrant instrument state microscopy, but this is just in anticipation of questions about whether we can use fluorescent proteins with STED. It is possible down to a lateral resolution of 80 nanometers. But of course, we prefer actually using the new technology of SNAP and HaloTag, using STED fluorophores as SNAP or Halo substrate to be able to do live microscopy with small organic fluorophores. And this is an excerpt from the publications of Francesca Botanelli, which she did in the Jim Hoffman group that was in Nature Communications in 2016. And there is also a talk of Francesca in an ABR Instruments summer symposium on our website describing what live STED is about and what we can do with it. So it's possible to do live STED using those methodologies. Now, to finish, a few words about MinFlux, which is the newborn of ABR Instruments. So MinFlux is a completely different technique. It's not STED anymore. This is very novel idea that was published by the group of Stefan Hell and imagined by Stefan Hell himself and developed in collaboration by his team, with his team. And they produced actually a method that gives nanometer resolution imaging and also single particle tracking of fluorescent molecule with minimal photon fluxes. So here the key thing is that we are going to be able to have localization precision at single molecule level, but at a resolution level that was absolutely unimaginable even 3 years ago. So it's really a single molecule localization microscopy technique that is using now a donut-shaped excitation beam, and which is going to be using much less photon counts to yield the same resolution that, say, conventional single molecule localization techniques such as STORM. So in a few words, how it does it is that we are using a sample very comparable to STORM with blinking fluorophores or, for example, photoconvertible proteins. And we bring the fluorophore in the on state. So the fluorophore is here, the yellow star. And when the fluorophore is in the on state, we actually cross his path by a confocal beam. Then as we cross his path, then the beam turns into a donut. The donut is going to be doing a circular trajectory around the fluorophore in the on state, collecting a number of photons. And an algorithm is going to be inferring where the position of the fluorophore was. And you can see, actually, as we are doing that motion, we are trying to stay as close as possible to the donut minimum. So we try never to be really close to the high regions of excitation intensity. So we really try to preserve the fluorophore as we're doing that and get it to emit as little photons as possible. Now, very interestingly, the dependency of the precision of localization is not anymore strictly on the number of photons emitted, but it now depends on the dimension of that pattern that the donut has done or on the dimension of the donut itself. And because we are able to tune that dimension arbitrarily, we can actually go really way down in resolution without actually being hindered by this quadratic dependency of excitation light and emitted photons. So with not so much light, we are able to localize with very high precision. That's the key message. And this is the kind of data that was published in the literature by the group of Stefan Hell on nuclear pores, yielding some precision of localization down to single nanometer numbers, like 2 nanometers in 2D, or even 3 by 3 by 3 in 3D MinFlux, which is something really completely incredible that was actually done in fixed cells, and now using even some constructs in mammalian cells, even in living cells, that was done on nuclear pores. So this is a complete revolution. And now the system is commercial since the end of last year. And as I said, this is a single molecule localization microscope that gives you less than 2 nanometers localization precision, even in the ABR instruments version. I mean, please, if you are really interested in acquiring that system, get your demo and you will see it's really true. There's no post-processing required, which is really important. You get the results straight out of the microscope. And in addition, this is ultra-fast. It has this ultra-fast beam scanning positioning. So it enables you to do the image not in an eternity. It has auto-alignment, but most of all, actually, that EOD-based beam scanning is enabling you to do single particle tracking at very high frequency. I think this, yes, 10 kilohertz is, it can be more than 10 kilohertz even in super resolution with mean flux. And the system is actually actively stabilized. So it's really a fully sort of a key in hand turnkey solution that gives you, in principle, 2 nanometers localization precision on an optimized sample, and not just in one color. It works in more than one color. We've been doing 2 colors and working on the 3rd color now, working on 3D in Flux 2. This is the kind of results that we obtain at Aber Instruments, and we don't need so much light to get good localization precision, and we can then push actually to 2 nanometer localization precision on this. So those were the spectrin rings, and now we start seeing single molecules of spectrin, which is really completely crazy. Same results, that's the nuclear pores. We used to see those, I can't remember, octamers or nullamers, and now we start seeing actually single molecule stainings in there with at 2 nanometer localization precision. Really, we see individual molecules, which is really insane. It's also a very nice tracking method at very high speed. We can track that lipid actually for 3 seconds. We have tens of thousands of tracks at super resolution. So I think it's somewhere between 20 and 40 nanometer resolution, or localization precision at least. And there are more and more papers coming up, and we've had our first delivery in Heidelberg. More systems will be delivered. So it's really happening. We now are able to produce super resolution at real molecular resolution. And with this, I want to thank you for your attention, and please get in touch if you are interested in seeing the systems in work.
Thank you so much, Frederick. It was such a nice and interesting lecture. I really like it. Now, I mean, we are running out of time, but we still have a few minutes if you want to ask any question. Otherwise, I would like to make you a question that a lot of people is asking, and it is about is it possible to, I mean, aesthetics coming on top of a confocal, or are there different systems? How is it working?
Yeah, no, they are not two different systems. In reality, the confocal is really built in. So we need to have a point scanning microscope enabled to do that. So our microscope is a point scanning microscope. It's not 2 separate machines. It's one single machine that does actually point scanning microscope and confocal microscopy and state microscopy. Yeah.
And some other question that many people is always asking, is it possible to have mean flux in 3D, or is just an XY measurement?
Yes. So of course, the images I've shown are really strictly 2D mean flux images. But Stefan Hell has been publishing an article about the development of 3D mean flux back in, actually that was in the beginning of this year, which was that was released. And I think I remember that the I can't remember if the Embod journal actually is describing 3D mean flux, but nevertheless, we are also developing 3D mean flux, and we are really close to releasing it. And we expect to obtain really a few nanometers in X, Y, and Z in 3D mean flux. So really close to the precision of localization of 2D mean flux. So we are doing it. We have many results now in 3D mean flux, and we are going to be releasing it sooner than later. Yeah, so 3D, yes.
Thank you so much, Frederick. Now is going to start some other sessions at the SPAUM. Then I would like just to thank all of you for joining today, and especially to Frederick to share with us all this nice lecture. And I would like just to remind you that tomorrow in the morning at 12, we are going to have the last talk from Nikon. That includes a live demo about artificial intelligence. Then I would like to invite all of you to come. And that's all for our side. Thank you so much.
Thanks a lot.